Good morning to our co-chairs and attendees of ISHLT 2022. It is truly an honor for me to introduce the recipient of this year's ISHLT Lifetime Achievement Award, my role model, mentor, colleague, and friend, Dr. Hannah Valantine. Many of you already know Hannah, but for those who don't, let me start with a brief biography. Hannah is originally from the Gambia, which my trivia-loving children tell me is the smallest country in mainland Africa, and one of only 2 countries in the world that start with the other one being the Bahamas. Hannah moved to London when her father was appointed as Gambian High Commissioner to the UK, which is where she pursued her higher education, studying biochemistry at Chelsea College, medicine at St. George's Hospital, where she was president of the Medical School Student Union, and then internal medicine training at London University, where she received the Sir Charles Clarke Prize for the student contributing most to the life of the school. Hannah then pursued cardiology training at Hammersmith Hospital, and I suspect that was not an easy path to take, given that there were only 2 other female cardiologists in the UK at the time. In 1985, Hannah first came to Stanford University for advanced training in cardiology as part of her BTA, or Been to America degree, as she often describes it, where she had the opportunity to work with Dr. Norman Shumway, a pioneer in the field of heart transplantation. Hannah's great potential was immediately recognized, and she was hired as assistant professor at Stanford in the mid-1980s and quickly moved up the academic ladder, achieving the rank of full professor in 2000, now over 20 years ago. Over the course of her illustrious career, Hannah has distinguished herself in many different and diverse arenas. First of all, Hannah's scientific contributions to the field of heart transplantation are tremendous. She started by describing echocardiographic findings that were associated with the development of acute rejection after heart transplantation. She then defined a causal role for cytomegalovirus in the development of cardiac allograft vasculopathy, and also demonstrated the utility of intravascular ultrasound for the early diagnosis of cardiac allograft vasculopathy. Hannah led several large projects funded by the National Institutes of Health and made significant contributions to our understanding of acute and chronic rejection after heart transplantation. Hannah then became involved in non-invasive biomarkers of heart transplant outcomes and led the first ever randomized controlled trial of non-invasive surveillance using gene expression profiling, the IMAGE study that was published in the New England Journal of Medicine in 2010. Hannah never rests on her laurels, as I have come to know, and is constantly thinking outside the box, coming up with creative ways to approach the many challenges that we continue to face in our field. It was around this time that Hannah heard of a non-invasive prenatal diagnostic assay developed by Dr. Stephen Quake, who was then professor of bioengineering at Stanford, that was able to identify and sequence cell-free DNA from the fetus in the mother's blood during pregnancy. Hannah immediately recognized the potential to translate this technology to the field of transplantation and asked Steve to meet her over coffee, and the rest is history. They pulled together a team of investigators who applied for and received a large NIH grant to develop a cell-free DNA assay for acute rejection monitoring after thoracic organ transplantation. Remarkably, less than 10 years later, the first clinical-grade cell-free DNA assay for acute rejection monitoring was made available to patients in the United States and beyond. Today, most heart transplant centers are using non-invasive assays to assess and monitor graft health after transplant, thanks to Dr. Valantine. In addition to her scientific contributions, Hannah has led the way in promoting diversity in the biomedical research workforce in the United States. Back at Stanford, she was appointed Senior Associate Dean for Diversity and Leadership in the School of Medicine, where she developed and promoted many programs to train and promote young faculty from diverse backgrounds. In 2010, she received the NIH Pathfinder Award to study the underrepresentation of women in biomedical faculty positions in the United States. In 2014, Hannah's career took a major turn when she was selected as the NIH's first chief officer for scientific workforce diversity, a new position that was focused on diversity in biomedicine. During her 6-year tenure at the NIH, Hannah's office awarded over $30 million in funding for outreach and career development work focused on members of demographic groups that are underrepresented in science. Under her leadership, the racial gap in rates of NIH applications and grants awarded to projects led by African American and Hispanic researchers has been greatly reduced. She also developed the NIH Distinguished Scholars Program, which aims to increase diversity among tenure-track investigators in the NIH intramural program, focusing on people of color, individuals with disabilities, and women. At the NIH, Hannah also successfully merged her work on diversity with her continued scientific work. She founded the Genomic Research Alliance for Transplantation, or the GRAFT Consortium, which is applying technologies such as cell-free DNA to understand the mechanisms underlying worse post-transplant outcomes in African American recipients. In 2020, Hannah returned to Stanford, where she continues to amaze me, constantly reinventing herself and pursuing new directions in her professional work. In her current role, Hannah is organizing large team science efforts, bringing together investigators from different disciplines to tackle important topics, such as the long-term effects of COVID-19 infection. Her work has been duly recognized at the national stage. In 2020, she was elected to the U.S. National Academy of Medicine, and last month she received the prestigious Pamela Douglas Leadership and Diversity Award from the American Heart Association. Throughout her career, Hannah has served as an amazing mentor and sponsor to so many ISHLT members from around the world, including Heather Ross and Nadia Giannetti from Canada, Luciano Potena from Italy, Tomoko Kato in Japan, and Mario Deng, Michael Pham, Sean Agbor-Ino, Palak Shah, and others here in the US. I have benefited tremendously from her guidance, and I still go to Hannah for advice about career and work-life integration. I've particularly admired Hannah as a successful working mother. She has 2 incredibly accomplished daughters. Erica is an MD PhD student at Stanford who is pursuing a career in orthopedic surgery, and Natasha is a student here in Boston at Harvard Business School. Finally, many of you have noticed that Hannah has an incredible sense of style. She is invariably the most colorful, fashion-forward, and vividly accessorized person in the room. Hannah stands out. She is noticed. In trying to figure out how she accomplishes this, I have learned that Hannah has a secret weapon: her husband, Dennis von Kepler. Dennis always has Hannah's back and is usually in the front row cheering her on as he is today. Dennis, please stand up so we can recognize you as well. I am still trying to figure out how I can hire Dennis to give me a makeover. And with that, I would like to thank Hannah for her many scientific contributions to the field of heart transplantation, for leading the way in promoting diversity in the academic workforce, and for mentoring generations of ISHLT members. I cannot think of anyone more deserving of the ISHLT 2022 Lifetime Achievement Award.
Thank you. That was amazing. Oh, wow. Thank you so much, Kiran, for that lovely, lovely outline of my career path. In fact, at this point, I feel that really there's no need to actually go into the talk, as you have told the audience so much. I must also just thank the ISHLT for this fantastic honor. I really feel so honored, so blessed, and quite frankly, humbled by this great award. I grew up in this society. My first meeting was in 1986, and I am sort of overwhelmed, really, by this honor, and I thank everybody for it. Let me just... So here are my disclosures, and I will not be discussing anything to do with off-label devices or any of that sort. So what I will be talking about is the many stories and one vision, that is the vision of improving the journey of our patients. And I will say that is what has driven me throughout my nearly 40 years of career in this space. And to do that, I'd like to take you through the lens of the work that I've done on acute rejection, allograft vasculopathy, cytomegalovirus, and very excitingly, in this new era of applying genomics to really better the lives of our patients. I'll start by a story of a patient of mine who's very dear to me. This is a patient who had a transplant at the age of 20, and despite advice from myself and Dr. Sharon Hunt to not get pregnant, she did get pregnant and gave birth to a very healthy child. That was in the early days of the immunosuppressive drugs, because we didn't know what it would do to the baby. Unfortunately, after several years, she developed a complication that we are all aware of called allograft vasculopathy. But thankfully, we were able to give her a new heart. And I'm delighted to say this was a selfie I took just last week in clinic with the patient. So you cannot possibly imagine the joy of having traveled through my career paths with patients like this. This is really one of the images that plagued me throughout my work, realizing that quietly and silently, people might be developing what you see on the coronary angiogram here, which is the allograft vasculopathy that often presented with sudden death. And throughout my career, I've committed myself to figure out what can we do to treat, to diagnose, to prevent, and treat this condition. So I think that I really do need to go back to my roots. When I arrived at Stanford in 1985, I was very blessed yet again by having the leader in the field of Doppler echocardiography, Liv Hatley, there doing a sabbatical at the same time. And it was working with her as well as John Schroeder, Sharon Hunt, and the wonderful Margaret Billingham, who's no longer with us, that I was able to look at the correlations of left ventricular diastolic dysfunction in the context of acute rejection, and develop a number of indices which we were able to track that were predictive of rejection, but somehow not sufficiently specific to replace the endomyocardial biopsy at the time. So with that in mind, I'd like to really chronologically talk to you about the work in allograft vasculopathy, particularly not only creating new preventive approaches and understanding the basic mechanisms, but really also the diagnostics. And what I won't be talking about is the rodent models that I developed. Yes, at some point I was a retologist as well, where we went to the lab and discovered many important things about the interaction of dyslipidemia and the allograft vasculopathy. But I think there was enough going on in the patients, which I can share with you now. And this was one of the very early studies where we were able to apply the use of intracoronary ultrasound when it was first developed by Fitzgerald and other people at Stanford. And with the transplant patients coming back on an annual basis, we were able to show that there was, in fact, indeed, intimal proliferation that was going on silently, even in patients that we did not realize. And we were able to show that degree of intimal thickening was predictive of outcome. And just think about it. What that meant was now we had a tool to track patients more sensitively, but also to assess the impact of our interventions on this intimal proliferation. And that was, I would say, a landmark discovery and a whole body of literature that emerged from that, which I don't have time to go through. But we did start to scratch at the surface of what was going on in terms of immunopathology. And here, I was very happy to have Mario Deng come and work with us as a fellow, where he was able to look at the correlation between the expression of particular cell surface antigens and the intimal proliferation, and in fact showed an inverse relationship, leading us to conclude that ultimately these markers might be important for us to follow. And of course, there was a lot of focus at that time on the importance of the metabolic abnormalities, in part driven by the tremendous landmark work of John Kobushigara and others showing that lipid-lowering agents were effective in preventing this devastating complication. And what I'm showing you here is some lovely work that was done by Tomoko Kato, who is here from Japan, while she was a fellow with us, indicating a distinct correlation between the intimal proliferation and the degree of diabetic control. Why do I say that that was important? It led us to be a lot more aggressive about the way we manage our patients in terms of controlling diabetes. And now, integral part of a lot of our programs are our endocrinologists who work very closely with us to manage these patients. So all of these are little steps in the patient journey to improve things for them. A very important issue came up very early on in this area of transplantation, which was cytomegalovirus, and the fact that it was when patients were immunosuppressed, we had at high risk of reactivating the virus and therefore developing acute disease. And so there was a real concern about how to prevent that. And in a wonderful randomized controlled trial that was done at Stanford, it was shown that the use of early ganciclovir for a month post-transplant diminished the likelihood of patients developing acute CMV disease. But in the back of our minds was the idea that the CMV was actually contributing to other deleterious outcomes, such as acute rejection and allograft vasculopathy. And there I was extremely fortunate, because in that randomized controlled trial that had been done, patients were followed up with serial coronary angiograms. And seizing on the opportunity of that to work with my mentor there, John Schroeder, we corralled these hundreds and hundreds of angiograms and had them reviewed, comparing the patients who were on placebo versus control. And the rest is history, where we saw that the patients who had received the ganciclovir were less likely to develop angiographic coronary disease. And there rose a whole question in the literature. Was, in fact, the CMV causal? And I was very fortunate to actually receive an NIH award to study exactly this. But it meant a multi-center study with a lot of hard work and a lot of coordination. And thank goodness I had the benefit of Helen Lucart, who is here in the audience, a nurse, a scientist, who helped us accomplish this work. And what we were able to show was, in fact, even though we treat these patients, there is a certain amount of CMV viremia ongoing, unbeknownst to us, that we called a subclinical, that could, in fact, be accounting for this problem. And it therefore wasn't surprising, ultimately, when we compared the patients who were having the more aggressive anti-CMV prophylaxis, that is to say, ganciclovir and CMV-IG, shown on the dotted line. Those patients, guess what, had less CMV viremia, and ultimately less rejection, and ultimately less allograft vasculopathy, making the case for Koch's postulate, whereby we had prevented something from an infection from occurring, and therefore presented the outcome as well. But I think one of the most exciting things of being in research is the fact that it is constantly changing, that those tools that you have for answering very difficult questions that come up in your patients every day when you go to the clinic are rapidly advancing. And this was actually what happened in the early 2000s when we began to have the application of new genomic tools. And here we had the gene expression profiling, which with the microarrays now became possible. And so working together with our industry collaborators at CareDx, a test was now available to see whether or not patients who were rejecting were immune activated through this gene expression profiling. And the rest is history. Michael Pham, one of our tremendous fellows now, a director of his own program, led this major randomized clinical trial which compared patients managed mainly by the gene expression profile or that by biopsy. And you can see the result was most actually fairly boring in that there was no difference. But what the huge benefit was that we saved a whole lot of biopsies in patients. And patients are very afraid of this. So this was really a huge advancement in the field. But when I saw that result, I've been continually plagued by the idea of what it is that affects the outcomes of Black patients. So in this study, where patients are very well controlled, I wanted to understand whether or not the outcomes were indeed very different comparing patients of different racial groups. Because we had seen in the kidney literature the idea that African American patients reject their organs more, and at one point it was thought that perhaps they were medication compliance. So in a well-managed sample in a clinical trial, it gave us an opportunity to begin to look at this. And you can see here, there's the event-free probability. So there's multiple endpoints, combined endpoints of graft failure and rejection in the white population in blue compared to the other groups, showing very clearly that outcomes are not the same. And this really has stimulated my thinking about what can be the underlying cause. Are we talking about genetic distance? Are we talking about mitochondrial DNA, given we know what the inheritance of mitochondrial DNA is and its propensity to serve as ancestry markers? Or is it simply related to the social determinants of health that result in such continual stress and ultimately stimulate immune pathways that lead to rejection. And as Karen nicely showed to you, all of these questions were really unanswerable until this current era of genomics. And serendipitously, I would say, sitting in my office one day and looking at the work that Steve Craig had done in using being able to identify fragments of DNA from the fetus in the maternal circulation, I thought this might actually work. And I called up Steve and we went for coffee, and this was his exact words, which I'll always remember. An organ transplant is essentially a genome transplant, so this will work. But he immediately said to me, Hannah, do you have any blood samples stored? And of course, we did, thanks to the great work that Helen Lukart had done in the previous program Project Grant. We were able to go immediately and rapidly to the proof of concept experiment. And the rest is history. From this work, we were able to show, yes, conceptually, you can pick up fragments of DNA from the donor out into the recipient, and that was the original PANS publication. We were able to also show that not only could you pick up human fragments of DNA, but you could also pick up bacterial and viral DNA, and it could ultimately be developed as a possible test for infection. And then, of course, as you all know, this was a small sample, and as we say in England, one swallow doesn't make a summer. We had to be confirmed in a larger trial, and we were very fortunate enough to get NIH funding to do that. And thanks to the tremendous work led by Karen Kush, who introduced me, we were able to move that from the conceptual into the actual patients, and she led a lot of these studies published on this paper. And then in 2014, Dr. Francis Collins connected with me and asked me to move to the NIH to serve as the first chief officer for scientific workforce diversity. And there my day job was obviously to begin to figure out new policies and programs that would enhance the diversity in the scientific workforce. And I'll get back to how that is linked to science. But also, if you know Dr. Collins, he's a geneticist, loves genomics, and wanted this person who was going to lead this effort to also be an active researcher from the point of view of credibility. It's one thing to, as an administrator, to tell other people what to do about diversity, but actually, if you're not living and breathing the science yourself, it becomes a little bit difficult. So I was very fortunate to have tremendous resources to do what I call my night job, which was the research part. And I got to NIH, and of course, I should have realized before getting there, there was no organ transplant program. There had been a kidney transplant program, but it was closed. So what was I to do? So what I did was I went knocking to the 5 local heart and lung transplant centers in the area, and I was extremely fortunate in that they all rallied around and were prepared to work with us into this program called the Genomic Research Alliance for Transplantation, or GRAFT. So it's a consortium of these programs here, including Hopkins, University of Maryland, VCU, Inova, and Washington Hospital, where they enrolled patients and collected samples in a very methodical way so that we could first see whether or not this cell-free DNA technology was reproducible. And I was very fortunate again there to have an outstanding physician scientist, Sean Agbor, who actually took the technology and made it really workable within the NIH. And here it is. It's a very simple idea. You take the plasma, you construct libraries, you do the shotgun sequencing here, and then you're able, through intuitive computing, to calculate the percent donor-derived DNA. And what Sean was able to do, in addition to driving this work, was to also show that it was highly effective for predicting outcomes in lung transplant patients, both allograft failure and CLAD, in this article that he ultimately published. But I think one of the pivotal trials, reports in the heart patients were co-authored by Sean and Palak Shah here in Circulation last year, which included over 170 patients. And I will say that 44% of those patients are African American. And so I think that, in addition to the validation study, is a major success of this trial, given what we know, the lack of representation in clinical trials. And this is just to briefly walk you through to show you, yes, the cell-free DNA was highly correlated with graft function as measured by ejection fraction. And in fact, as you went up higher grades of rejection, you have more, higher percentage of donor-derived cell-free DNA. And in particular, notice the orange block on the right side, that is antibody-mediated rejection. And we really believe that both from the quantitation and the characteristics of the cell-free DNA, we now have a test that is highly predictive of antibody-mediated rejection, which after all is the Achilles heel of transplantation. And as you've classically seen in these publications, here is the data from the heart patients, and similarly the data from the lung patients showing this correlation. And when we look at the receiver operating characteristics, we see that it's highly predictive. And so now we are able to move it fully into the clinic. And in fact, at Stanford, these days, patients after the first month are not getting any biopsies unless indicated, as you will see in many of the presentations in this meeting. One of the key things that we noted was that the cell-free DNA is increased way before the heart biopsy, and that might have been contributed to the false positives that so many other non-invasive tests have come up with. And this is just a diagram to show you how it is that in this set of patients, we see that the cell-free DNA elevated up to 3 months earlier than the biopsy. So I think we are on firm footing now that this is a test that we can use for our patients and diminish the problems of biopsy. I've already mentioned that this graft cohort that we developed from the patients in the Washington, D.C. area was very diverse, and here the representation of the different races and women. So that really, again, puts us on a firm foundation to begin to look at and answer the question why the differences in outcomes. And one of the early work that we saw here represented on the top is, yes, unfortunately and sadly, yet again, we're seeing this divergence in outcome comparing the black patients versus the white patients in this graft cohort. But if you look at their levels of their cell-free DNA here with the black patients represented in red, what you see is that the levels are consistently higher all the way through after the transplant. And what that means to me is that there is persisting injury that is occurring in the graft, and this is despite equivalent levels of the key immunosuppressive drugs. And so this clearly busts that old stereotype that people are not taking their medications. But even more importantly was the observation that at the first day post-transplant, 24 hours, the levels are higher. And so I couldn't help asking the question, what cell-free DNA? Is it a donor cell-free DNA? Is it total cell-free DNA? And for that matter, is it mitochondrial DNA which becomes quite relevant vis-a-vis the previous talk that you had. And when we dig deep into it, what we're seeing is that, yes, the patients who are African American have indeed higher levels of the cell-free DNA shown on this slide, and I'll move pretty rapidly. And this is all preliminary data. We're now seeing that, in fact, both mitochondrial DNA and nuclear DNA are significantly elevated, but in fact, the most important situation where we find this elevated is in the context of antibody-mediated rejection. And this leads us to a study that we did asking the question whether mitochondrial DNA and whether mitochondrial DNA mismatch between donor and recipient can predict this rejection. And all of the preliminary data is pointing to that. So look to that for the future. So let me just close by saying I walked through a lot of what I've done in the last 30 years in terms of the patient journey and making the diagnosis of rejection easier so that we can have better outcomes for our patients. But at a higher level, I believe that where science is going is that every scientist, every researcher, and actually every clinician needs to be asking themselves 4 questions about the work that they're doing. And this is an article that I've written published in Nature with some colleagues at the Chan Zuckerberg Initiative, where we say the first question to ask is, who is doing the science? Who is in your research group? And the reason to ask that is that those people bring a diversity of thought and they bring new questions to the research agenda. What science is being done? What is the kinds of topics that are being investigated? And are we looking at the outcomes by sex and race, etc.? And then thirdly, how is science being conducted? And what is the technology being developed? And when these technologies are not tried and tested across different sexes, across different genders, and across different races, ultimately, they're not going to be widely usable. A classic example being the pulse oximeter that was under-reading the O2 saturation in Black patients. And then finally, who has access to all of these wonderful products of science? So I'll close by acknowledging the great lab that I had at NIH, which allowed me to continue the work that had begun in Stanford. And key in that lab was Dr. Sean Agbar, who is now a leader in genomics in lung transplantation. And that wonderful team of all of the investigators at the 5 different centers who actually worked with me. And then finally, to my wonderful family, Erica, Natasha, and Dennis, without whom none of this would be possible. And again, thank you very much to the organization for this prestigious award.